All right, the stage is yours. Thank you, Vince.
An ecosystem aquarium. Why is it called that here, you ask?
Well, thank you so much, Ludwig, for an absolutely flattering introduction, and I really appreciate it. And it's a pleasure to talk in this seminar series, Computational Biomedicine. I'm going to discuss some fairly general concepts, and I think, let's see how we do this here. The board is not moving. We've now moved to another board. How are we doing? How am I doing? Anyone have any thoughts about this here? Here we go. Now it's not, there it goes, it moved. Okay. Just what you wanted to hear, right?
That is
So
Zoom.
here we go. Here's an overview. Everything looking good now?
Everything's looking great, Vince.
Oh, thank you. I need a little feedback every so often.
Certainly.
So I'm going to start out with a little bit of a snippet from this open microscopy environment project. And I'm going to use that as sort of a motivation for thinking about this thing very generally, an open computational ecosystem. And then we'll start asking, what are the components? And we'll think about GA4GH standards as helping us to think about the most prominent components that we should be working on. And then we'll look at this Anvil NHGRI cloud-based system in relation to GA4GH and think in terms of stacks of software that are involved in Bioconductor. And then I'll conclude with some things related to Kubernetes applications, which I think ultimately move us in the direction that we need to go in, which is using containers and reducing the amount of technical work that users have to go through in order to get a computational ecosystem that they can work with. And that's it. So it's a lot of material. I may be very obscure in certain places. Please just interrupt me. Just call out. So I found this little piece of discussion in connection with the open microscopy environment. And let's just read a couple of the lines here. Bioimaging science is at a crossroads. Acquire more, larger, preciser spatial measurements is at odds with our ability to structure and share those measurements with others. Bioimaging data should be shareable via open and commercial cloud resources without the need to download entire data sets. This is not the norm. The plethora of data formats produced by imaging systems are ill-suited to remote sharing. Individual scientists lack the infrastructure they need to host these data themselves. And so this is thinking about a file format that may combat these shortcomings of current computational approaches to bioimaging. So the way I read this is that as biotechnological innovation keeps roaring along, and we have these strong ambitions in biology and clinical applications, we're going to run into a lot of platforms that are going to produce these high volume data, digital data, imaging data, et cetera. And there's a lot of variation in computational resources that can be used. And the research community can't really be effectively mobilized. There's just too much going on that's of a clerical nature, downloading data, reformatting it, and everyone's doing it themselves in silos. One of the most striking things I heard recently, talking about a large group doing GWAS, is that we won't use the new reference genomes, even HG38, until most collaborating research groups do, because then we'll be out on our own. We have to stay in sync. And so, this open microscopy response is, well, let's build some consensus for formatting the data, layouts and metadata. And similar things are going on in other areas, of course. So we wish them well. We hope that these standard formats come about and that we're able to use them. And just what we need in order to use them in the best way, maybe, is this discussion of an open computational ecosystem. In other words, if we knew how to do the job with image formatting or matrix formatting, we'd be on our way to maybe coming up with consensus on other approaches like storage. So once you get the format down, you have these terabyte sized things. Where do you put them? And how do you manage them in a safe way? And how do you allow people to interrogate them? Computing. So there's CPUs, GPUs, and orchestration of clusters. Infrastructure, so low-level communication, scaling, container management, CI/CD for those who are writing software, and instrumentation. I'll spend a fair amount of time on that. How do you know whether you're using your resources in an efficient way? Tools, so that's where we've spent a lot of our time in Bioconductor. Ludwig cites 2,000 packages. 2,000 packages that people need to know about in order to find the tool that they want to use. So these tools are many, and their characterization is often somewhat vague, and they are built into workflows. So they're parts of workflows. They all need to be benchmarked. They need to be validated. They need to be maintained. And that'll certainly be an important part of the computational ecosystem. Reference data is something that I think Bioconductor has done a really great job on, and it's a very humdrum topic. I mean, how hard is it for you to get the whole sequence of any genome for any organism you're interested in working on? And which version of it? And how about the transcriptomes derived from them and so on? Well, Bioconductor has done a good bit of work on making that all work for people who are R programmers and potentially more generally. I don't think that this issue of making sure the reference data are in the computational ecosystem that you're working with is a solved problem in any general way. Well, even more clerical in nature are authentication and authorization, general security concerns for working with the type of data that you'll run into if you're working with human genomics. And this is something which you cannot get around, and is certainly an obstacle to folks who want to use clouds to do really big computations. They have to be able to understand and use all of this. And GA4GH, I think, is coming up with solutions here that should be deployable soon. Well, that's all very nice. How do you get people to buy in and work together? So I call that socialization for evolution, because whatever you do, you're going to have to change. And you're going to have to change it in ways that make people happy. And since people are, busy with things they have to do, gathering requirements and consensus on, when we can make a change. And then the governance of the community resources that come into play when you have a federated ecosystem. This is something we have to have a good approach to. Slack is pretty good. Email, not so good. Other things exist, and I'm not really up on them, but metrics for how you socialize, code of conduct, health of the ecosystem, and so on. These are all important things. And of course, I've forgotten a lot. So these are the types of things I'd like to have us sort of get our heads around in conjunction with this topic. And so GA4GH seems like a pretty good place to go. Lots of very energetic and expert individuals building this system of standards. And the biggest topics are genomics, regulatory, and data security, all things that I've mentioned in some sense here. And one of the components that I don't know that much about, but seems like a really good one, is a service registry API. So you can actually identify all the services that you think are relevant. So Beacon Network is an interesting one. If somebody finds a variant that has never been seen before, they can go and Beacon and let people know that this variant exists, and other people can look for it, and we can start to get a sense of its prevalence. Workflow execution service. How do you make a workflow that people may think is really the best in field? How do you make it accessible to people without a great deal of effort on their end? And even search. So these are services that at a very general level, it's great to be able to tap into, if you know how to mobilize these GA4GH components. Well, I sometimes think of this anvil that I have done some work on, thanks to the vision of James Anthony. James, I'm sorry, I'm blocking on his name.
James
Taylor.
James Taylor, yes, thank you. James Taylor, who brought us into this anvil business, where there's a Google Cloud platform based on the Broad's Terra, and then we are working on this to bring Bioconductor into this notion of inverting the model of genomics data sharing with this Anvil platform. And this has its own set of modules. So Gen 3 is a genomics data cloud infrastructure with data models, indexing and querying, workspaces in Terra, DocStore for this tool registry service, that's a GA4GH standard, Jupyter to do interactive computing, Galaxy for workflows in an interactive environment, and Bioconductor for analysis and comprehension of genomic data interactively. So these components represent the Anvil. And you can see here that a great many terabytes of data from very important consortia have been assembled here, first through Gen 3, then accessible through Anvil workspaces, and then all of these other components can be used to tackle the analysis of these resources. So to summarize to this point, this concept of an open ecosystem for modern genomics must be formulated in a rapidly changing scientific technical context. And I find the GA4GH specifications very useful for getting a handle on scope. And then if you want to implement GA4GH, well, Anvil shows you a way of doing this. A few of these things are online now. And I'll show you a little bit about where we are with respect to this data repository service, which is going to mediate in a secure way between users and data that they need to be authorized to use. And that's through this thing called the Researcher Auth Service. And that's an NIH-oriented initiative. And there's also this thing called Cloud Platform Interoperability. All of this stuff is knitting together in the context of Anvil and some other large platforms. And so this inverted model of data sharing is very nice to think about, but it's still quite a ways to go to carry it out. So I'll stop there to see if there are any questions. I see something in the chat. I don't know if it's, yes, welcome, that's good. Any questions or comments to this point? Because I'm going to transition from these very general considerations to more details about Bioconductor. All right. So this is a little schema that I put together when I was going to teach a course that got canceled to introduce people who are mostly genome biologists to Bioconductor. And to explain to them that while they are, clearly very expert here and probably doing things in systems biology, they have to work through biotechnological productions of sequencers and single cell and genome editing and so forth. They'll get all this digital stuff, and eventually they're going to have to do some high dimensional data science on it. They want to get from here to here, but ultimately they have to get up to here, which is where they're actually doing the biological inferences. And in order to do that, Bioconductor is awfully useful because these digital outputs can be pre-processed and analyzed in a nice way that makes use of modern data science and modern statistics and so forth, and that has been going on for quite some time and seems to be able to keep on the cutting edge. So that was a motivation to give them a sense of what are the different waypoints you need to get to in order to do the things you're going to do in this course. And I find this schema still relatively useful in getting someone from zero to, okay, I see what Bioconductor may be capable of. But it doesn't really get to where it needs to be because all kinds of new things are coming in here. Data in our experiment hub and our annotation hub, reference in the annotation hub, and lots of code in the software packages that people contribute. That's coming in all the time. And then going out are the training resources that are fundamental in all the packages that are in Bioconductor the vignettes for all the packages, the authoring that is given, and then these ideas of actual interactive training that Sean Davis has helped us implement with this orchestra system that I'll talk about a little bit later on. And then there's the issue of discovering things and the opportunities given through this ecosystem, let's say. So 20 years we've been doing that, and now it's not going again. So we've got to just figure out where we are. Okay, there we go. I don't know why it slows down like that. So here's introspection that I think we need to do very significantly. What families of components drive the value of bioconductor to genome biology? And I'm going to suggest that there are 5. The analyst stack, that's the one that everybody's really thinking about when they say, oh, you have 2,000 packages. So the analyst stack is supposed to be secure, reliable, and efficient tooling for genomic data analysis. And we pushed that a little bit further because we used to provision virtual machines in Amazon. And now we make these containers that really take the analyst stack and make it very easy to get it going without having to configure your Mac or your Linux box or your Windows box, whatever. You just get a container and you go. There's also deployment of workflows supported and clear paths to data, right? So that's what I consider to be the analyst stack, and we do a fair amount of it. The developer stack is, well, how do you write a new piece of code that you think people are going to be able to run with for the analysis that you think is appropriate for this new modality? And for that, you need data structures. I'll talk a little bit about that, annotation, and capability to benchmark, to compare your techniques to those that are already out in the field. You need source code control and good paths to contributing into an ecosystem that is open and viable. In addition to this, there's a documenter stack, right? It's not enough just to write code. You have to actually write a manual, and you have to write vignettes that help people really understand what's going on and how it gets used. And that's really important for workforce development as well. So 3 stacks already, and they're all kind of working out okay. Then there's the teacher stack. So what's the best way of getting material into a student's head? Well, we have workshop authoring and live deployments to students. I'll tell you a little bit about that towards the end. So this is all wonderful. And, you know, it was all created, or a lot of it was created before GitHub, before Git. And nowadays, you know, a lot of people would think, well, you have a bunch of Git repos or GitHub repos, and, you know, a lot of this can be accomplished there. But there's the deep stack. There's something that unifies the management and evolution of all of these things as R is bumping along through its versions, and as bioconductor packages are tracking, and as new people are throwing new things in there. There's the deep stack. I'm not going to say much about this, but it is something that makes all of this available to people, and it needs its own life support. So here's a view of the analyst stack. Linux, 4,322 packages. So 2,000 are bioconductor, another 2,000 are CRAN packages that they may depend on. Lots of packages. And this is 3.14, so this is going to be frozen pretty soon. And you can see the status is that the majority of them are pretty well in green state. That means they're passing check. All their unit tests, if they should be sufficient, are there to get them to a reliable state. So I would say this is a pretty good situation for bioconductor packages in this ecosystem. And you find things using this ontology made 20 years ago called BioC Views that gets updated every so often. If you have a biological question, let's say, related to the 3D structure of DNA, then there are 7 packages that may help you out. Likewise, 139 for gene set enrichment. So you have to go through and take a look at these, maybe benchmark them, and then you decide how you're going to go forward with your analysis. Well, that's great. Here's single cell experiment. This is a new data container for single cell biology. And it turns out that having created this a few years ago, that team is now supporting 115 packages that depend on it. 116 maybe. And you can get information on their state by looking at this page. So this is part of the deep stack, telling people, well, your package here, which depends on this one or gives things to this one, is working or not. And it helps you figure out, do you want to make a change? Or if you do make a change, what could its ramifications be? Questions or comments to this point? We wanted to leave some time for discussion. I think we're doing pretty well. Okay. Well, on we go. The developer's stack. We talked about the developer stack a little bit so far. Now we're going to say, if you're going to write some code, here's what you should use. So this is a schema for the multi-assay experiment. So we had something called a summarized experiment, which takes an array, let's say, of feature by sample quantifications, and has all the metadata about the features and all the metadata about the samples all unified together very nicely. You can use it as if it was an R matrix and make all kinds of interrogations that are very helpful. Well, we have a multi-assay experiment then, which generalizes that to a larger number of assays. And the initial use case was TCGA, where each tumor type has a whole cohort of individuals, but also expression data and mutation and copy number variants and some proteomics and so on. And so linking them all together in the same way so that you can think of these as a holistic entity for which you can do certain types of bracketed subscripting has been very productive. It really helps people get running with TCGA in very little time. So this initial use case, I think, has worked. And my questions are, can we go further? In other words, we can do TCGA with one data structure, but what about something more complicated? And here you might think of some of these things like All of Us, or the Million Veterans Project, or Human Cell Atlas, or this Human Biomolecular Atlas Project. Yeah. T2T is another matter, right? That's a different reference genome. But again, you want to have a holistic representation of that to help people get involved, to help them interrogate it. Should we do this? And how will we know if it's done well? These are the problems of an open computational ecosystem. And so I'm going to try and demonstrate this with GTEx. So GTEx, you could say, well, you know, it's not that much bigger than TCGA. Maybe it's not such a big thing. So let's look. This is how GTEx portal is described. It's an ongoing effort to build a comprehensive public resource to study tissue-specific gene expression and regulation. 54 non-disease tissue sites across 1,000 individuals, primarily for molecular assays, including whole genome sequencing. Hold on a minute. Okay. Exome sequencing, et cetera. Oh, there it goes. All right. So here we go. I'm going to use a multi-assay experiment to deal with the GTEx structure. And here I'm not really looking at it in the sense of a multi-assay experiment where there are multiple genomic assays applied to a given sample, although that is certainly the case here. But I'm just going to use the multi-assay experiment to deal with the different tissues that are in the GTEx. Okay, so there's a package called curated Anvil data, and it has a multi-assay experiment that represents GTEx. And if you look at the number of columns, ignore the fact that we have 0 rows here. That's part of the abuse. But the numbers of columns are really very substantial here. And the way we get that is that this project, as represented in Anvil, has many thousands of transcriptome profiling results, different types of results, aligned reads, ASE, junctions, unaligned reads, and so forth, that can be available. That's the RNA-seq. There's also whole exome and whole genome. And I wouldn't make too much of these zeros here. This is sort of midstream. And so we want to program with this. I've got this thing here, which represents, in my view, all of GTEx. But we have all this GA4GH capability to think of this through a data repository service. And so we can start programming on this. We can say, well, I'm interested in the lung. And I'm going to just look at the call data and find those elements of the call data that happen to have a bigwig format. So that's sample level data. And now I'm going to look at this variable called pathology notes, PRC. and start looking around for, are there any interesting clinical features that were noted in these samples? And I was kind of surprised that these are healthy people, but we find things like fibrosis or emphysema and so on. And in fact, I did a little bit of text mining there to just see what are the most prevalent conditions found in this corpus of terms related to the different samples. And it turned out that there was, quite a bit noted there. And emphysema and fibrosis seem very noteworthy. So if you're thinking of GTEx as a place where you go to get the normal relationship between genotype and expression in healthy tissues, you might want to know this type of information. And so the upshots here are that this ANVIL data model for GTEx can be put into a multi-assay experiment. And that's very much thanks to Martin Morgan, who wrote the ANVIL package that wraps up all of the Terra microservices so that you can call them with R functions and they work beautifully. And then this use case on investigating clinical notations on pathologic characteristics of samples was applied, and it's easily resolved. Now, what about getting to the assay data? That's where DIRS comes in. And this is, again, midstream. This is not the mature approach to this, but this is more prototypical. But we have these DIRS URIs, Data Repository Service, URI, which is a canonical representation through this standard of some data on the lung. And it resolves to this URL here. That's what that thing is. And now we can process this using the microservices to start getting the assay data. And here I can just pick a place and get the pile up to see what's expressed. So that seemed pretty nice. And here's another one. So there's this thing about GTEx where there's an ischemic time. People have died. We're going to harvest their organs. How long did it take between their death and the harvesting? And if it's very long, we might want to know and maybe see whether those samples are any different. And so we're able to filter the data according to this and see that the thousands of columns now go down that we had previously, right? Because this condition is not very frequently met. But when it is met, we'd like to know. Okay, so this type of programming, I think, is very straightforward for bioconductor folks, and it can be applied to these very significant consortia level data that get there. So the moral of the story is what's worked with us in bioconductor and R for 20 years of file handling, being efficient with respect to how we use RAM, and keeping the metadata tightly bound to the data. It continues to work when the data and the annotation are acquired through these managed, highly privacy vetted microservices. In this case, Terra is FedRAMP compliant. So it's really a very secure place to be as long as these protocols are being followed. And so we can hand off the authentication of authorization to the hosting platform, which is going to have RAS, and RAS passports will be used to see whether you can actually go and get something out of that DIRS resource. And then big distributed workflows are the next thing. We've shown that you can represent the data this way. How about doing these big jobs? Well, of course, you can use Cromwell or whatever else they have in there. But it seems to me that if you're thinking about GA4GH and you have bioconductor representations like genomic files or multi-assay experiment plus BioC parallel plus Kubernetes, you can orchestrate these all together and use a familiar programming environment to do things that are really large scale. Well, the question that leads to is, should we be looking a little more at the coherence and perspicuousness, right? If I can think of GTEx like that, that might be very valuable. And so I think we can. I don't know how many people are going to jump on board, but with enough demonstrations and what I call a youthful spirit, we should be able to do that.
Vince, what do you mean with just make sure that S4 vectors is working well? Isn't it working well?
Well, all I'm saying here is that S4 vectors is kind of the deep piece of software that that makes all that stuff work in a certain sense. And thank you for asking that, because what I meant here is that you really want this thing and other pieces of software that are at its level to be highly, highly unit tested and broken and then rebuilt. You want to know every vulnerability that there is in this stack. And I don't think we have the time or motivation to do as much as we should be doing in the reliability space with respect to the things that make this work. So that's my caveat. That's my claim that's my sort of cry for investment. It's a community service. It's a community open ecosystem. And this might well be one of the most important comments in the whole slide deck. Because if you aren't really committed to engineering the pieces that you're working with, you probably shouldn't do it.
And Vince, yeah,
yeah.
go ahead. No, I just say we have another question in the chat, and I'm happy that a native speaker asked that and not me, who is a non-native speaker. He asked, Vince, can you give a definition? This question comes from Sean Davis of let me see whether I can pronounce this, perspicuousness.
Perspicuousness. Well, maybe I forgot what perspicuous means. I think it means easily surveyable.
I see.
So you get a sense of the whole big picture. There's perspicacity. That's a person who sees the big picture. Perspicuous is something which represents the big picture. Maybe I'm wrong about that. It's an old 10th grade vocabulary word, but I hope that's helpful.
Maybe a more thematic question. I hope I'm not taking away time from the talk, but you asked for questions. So I saw this T2T and this telomere to telomere, which are claiming, okay, we are now having finished the complete genome, especially all these highly repetitive regions. And I just wondered, what's your take on that? Is that something real? Is it just a paper, or do these folks have the authority to make this another reference assembly, such as HG19 or HG38? Are we going to see that coming and using?
Oh, yeah. Yeah. Great point, Ludwig. This is noteworthy to me because it was done on the anvil, but it is real. And it is a prototype of a method for doing more accurate genome sequencing at the deepest level of resolution that will then be applied for personalized genomics, right? So I think there's a lot going on there. Is it going to be HG40 or something like that? That I don't know. But I think it probably could be. And I don't know the exact path forward. But there's a whole slew of papers that were just released recently about that technology and its results. And so, yeah, I'm not, I think it's important because it demonstrates the capabilities of the anvil. And it's something that's going to mean a lot more work for us as we try to internalize that level of annotation into our system.
Interesting.
Okay, let us continue. Let's see where we are, 37. I think we're in good shape. Were there any other questions? Okay. Again, when I pause, it starts to get, I think our little guy is not doing well. Zoom, Mac, and Google, or whatever this is. Here we are, point to ponder. So now I'm going to transition from sort of data structures and bioconductor to infrastructure and making sure that people get what they need. So a happy bioconductor uses BioC Manager install a lot. I certainly do. But of course, I'm reviewing all the packages that come into the system. So I wind up using it a lot. But if you're exploring new methods, you're going to install something new, and then that's going to require some other installations. And ultimately, you're going to bump into something for which you don't have the right runtime. Like if you're trying to do spatial stuff with SF, and then you need GeoJSON IO, and you need GDAL, and so on, we bump into these things. And it uses the internet and local compilers to get a new package and all its dependencies. And this can frequently fail. And for some of us, it's like, okay, yeah, I'll go and straighten that out. But for a lot of people, it's like, nope, we're not going there. And that means I abandon a project, or I get a new laptop, or I work a lot to upgrade the machine. And containerization helps avoid this, but it brings new requirements, certainly to us and certainly to users, and some vulnerabilities. And I'm not going to get into that. But I think the software stack and how volatile it is, how frequently it's updated. Let's say R. R was used to be every 6 months. Now it's kind of a year, but there are point releases anytime. When do I have to really make a change in order to respect that? Can that level of volatility be reduced? Can periodic release of containers coupled with package repositories, which really help things work, when do we know that it's sufficient for the majority of research requirements that are out there? We're not there yet. I think we can make it very easy to use. We can watch RStudio, who have these wonderful binary repositories out there. But this is something which we haven't been involved with for a long time, and we're learning how to do it. So binary repositories are important for Anvil, which is already container-based, and they have other roles of value. For distributed parallelization, if you have a bunch of workers out there, but they don't have exactly what they need, they can either sit there and compile it, or they can actually get a binary that they know is going to work. And that's accomplished through this. And in 3.15, users of GitHub Actions for their packages via BioC will not find the build time being consumed by compilations of package sources, because the binary repo will supply exactly the things that the container needs in order to install with 3.15. So it's my view that learning to produce and manage binary repositories of this software stack is a big step towards realizing this goal of an open computational ecosystem. But we have to analyze this process. Someone's going to pay for this work, where you're building these binary repos for 2,000 packages every couple of days. Oh, looks like we might lose our power here. I'm going to just keep going. So this is a nice little story. This is CPU utilization and this is due to Nitesh Turaga and Alex Mahmoud, who have put together the whole system for doing this. What happens is you start out building packages that have very few dependencies, and that goes very fast. You can use everything. And then you start hitting these packages that have lots of dependencies, so they have to wait until all the dependencies are built, and then you can get more utilization. There's an interesting analysis there. And timing this and building this so that it works and uses the computer that you've paid for seems to be a very viable thing and very valuable to do. So that type of analysis is important. This is memory usage. So this is free memory, and you want to make sure that you aren't over provisioning the systems that you're working with. And it looks like we might be to a certain extent here. But there's granularity. And finally, disk usage. As you go and build these guys, new disk gets used, and we see that. So scalable repository production is working, but it needs to be analyzed in order to make sure it's efficient. I'm going to close out with some discussion of this orchestra system that was hatched by Sean Davis. This is the 2021 Bioconductor Workshop Series. So all of these wonderful topics have been contributed by people who not only give you the text of their courses, but Docker containers that we know will run all of the code. The source code is there. And then when it's time for you to give this to 20 students, you tell them, well, go to this URL, you'll get a username, etc., and everything will work for you, and you can work along with us. And that is quite a nice system for authoring and deploying training material. And then Alex showed me how you can use Galaxy's monitoring system underneath this in order to find out what jobs are being run and whether students are doing what they're supposed to be doing. So that seemed interesting to me. I haven't really spent any time with that. And so to conclude, I guess the question is, do we agree about any of this? Are these the components of a vision of a computational ecosystem that is open? Have I told you enough about storage, compute, infrastructure, tools, reference data, authentication, socialization? In the context of Bioconductor and the way we're pursuing some of these things, I haven't mentioned, for example, Open Storage Network, which we're working with to really make large-scale data more accessible to people. And it's an NSF system that they've been somewhat generous with us to give us space there. And then the ways in which we're also going to use Jetstream or Exceed to give people time on computers to use GPUs to build and test the packages that are now being contributed to Bioconductor that use GPU to do their genomic analysis and machine learning. A lot of things going on that I think are components of this ecosystem that we want to have, and that Bioconductor is taking steps to help us have. So I'm going to stop there and hope that my battery doesn't run out before 4 o'clock. Any questions or comments?
Yeah, thank you so much, Vince. Perfectly in time. Great talk. Thank you very much. I wanted to ask the audience I have 2-3 questions, but maybe I check in with the audience first. Any question for Vince? You can either just like raise a hand or just speak up, unmute yourself, or use the chat, whatever you prefer.
Well, I see a question from Nathan Palmer, who has a question about binary repository maintenance.
Mm-hmm.
Yeah, so it's interesting. I have done sort of a poor man's version of what you guys are doing in a much more rigorous way to maintain a set of Docker containers that are sort of purpose-built analytic platforms for a variety of epidemiological research coupled to health services data. And the question of compile times, although not nearly on the scale where you guys are at, has come across my radar a bunch of times. And one thing I've been thinking about as a computer scientist for a long time is, are there any tools available in the R ecosystem that can help us understand the dependency graph of some of these things and parallelize the compilation of these packages, right? Because it's frustrating to me, even on my stupid little laptop, I have 8 cores, right? And only one of them is active for most of the compilation time. I'm just using a standard GNU toolchain. But I'm interested, as you guys are thinking about in actual dollars, what it's costing to rent these instances in AWS or wherever you're running them. The need to keep the CPU as hot as possible is real. So I'm curious as to whether you guys are putting thought into that and are actually doing anything in that space.
First of all, if you're talking about R packages, then if you used ncpus on your install.packages calls, you would find that it would parallelize and it would use make -j to figure out what it's going to do. So yes, there's lots of stuff available to do dependency graphs among R packages, and we are using it, and that's how I got that that initial plateau that we saw there here. So what we do, we start out with the packages that have essentially no dependencies. They can all be done in parallel, and that goes on for quite some time. And then you start to have dependencies where people have to wait for other packages to come in. So there's some stuff there, and I think built into R is the capability to parallelize installations. Federico Marini, thank you. In the chat, he's shown how to do it. So for other more general problems, I'm not so sure, but make might be able to help you if you're using that.
Excellent. Other questions for Vince from the audience? If not, I wanted to ask 1-2 things, Vince. It was quite interesting for me to see that you were starting off your talk with OME, next generation file format, and that has been actually something that we have touched quite a bit on in the last 2-3 seminars on spatial transcriptomics, and also in offline discussion with Niels Ehling, with Dario Righelli on spatial experiments, but also the SquidPy folks. Are you aware of any adoptions yet? So I understand, okay, they're basically like discussing these 3 file formats. I was just reading this paper on the weekend, so it's pretty fresh. An interconversion between TIFFs and HDF5 and ZAR. Are you aware of any adoption within R and Bioconductor yet for this? That seems to be a pretty hot topic.
Personally, no. I picked it up because the text that they were narrating there was so connected to what I'm trying to think about here. So it was very convenient. But I came across that OME through the FOM, this feature observation matrix that Ambrose Carr is spearheading through the CZI for single cell. So I think this idea is very hot, but I don't know where it's being picked up in terms of the types of software problems that we're facing at the moment.
Mm. Yeah, it looks like we have become pretty good with these large matrices, right? With HDF5, and now these images are posing a whole set of new challenges, and they're putting forward the ZAR format. And in Python, they seem to be already a bit further ahead and have already a native ZAR reader for that. And I think there's some discussion for adopting something of this for R as well. So it will be quite interesting to know what's the status there.
Yep.
All right, and then on the other hand, you mentioned these annotation resources and reference experimental data sets, and we are kind of like underestimating how valuable they actually are, and we kind of like take them for granted in R and bioconductor. And if when we recently did some work in Python, we were kind of like lost because we don't really have them, and I wondered how difficult it would be to like open them up like for other ecosystems as well, given that there is quite some cost to it, I guess, to like pulling these down also programmatically, not only within bioconductor.
Yes, great point. So there are two things there. The first is putting all the reference data in Amazon Open Data. That will be there. And then there's another bit of work going on with, this is going on with Nathan Sheffield, who has done this thing called BedBase. So he's very interested in language agnostic provision of reference annotation, mostly in the bed world, bed files. And he has an API for that. And I think we're going to be trying to combine that with a revision to the Annotation Hub infrastructure in the not too distant future. So I think if we knew your Python use cases that you were having trouble with, it'd be interesting to see where the gaps are and whether they can be met. If our data aren't in RDA format, and they often are not, you might be able to use them directly. Otherwise, we need to see which things are high value that should not be in RDA format, but it should be in something more language agnostic.
Oh, that starts with some very basic things such as a OrgDB package for human or a TXDB package for HD38. These are the.
Well, that's where I would say, thinking about them in their deeper representation, which is as SQLite databases, you should be able to work with the internals.
Mm-hmm. Mm-hmm. Mm-hmm.
Then we need to write maybe some Python code to make it as easy to use as the TXDB type structure. But the deeper data, I think, is all language agnostic.
Mm-hmm. All right, great. So any other question for Vince? I saw Tyrone was raising a hand. Is this still a question?
It's sort of a very general question, just for a lot of the things done earlier. I didn't want to drag on with to avoid getting the technical stuff right here, which is very fascinating. It's just a lot of the issues for very ambitious open data frame right there oftentimes build on consensus. The only thing is that I noticed working in commercial program development, most of this is relatively brushed as water under the bridge, mostly not because there's no vision necessarily, but it's just a lot of the times development is spent on short-term goals. You have to, especially the agile development, which asks you in order to bring in concrete artifacts on a regular basis. So there's not really a lot of time to do a lot of these things when you're trying to rush up a project for your next sprint. So I wanted to pose a question, maybe as a general philosophy. Do you see any ways to hopefully bring down the timescales on a lot of these issues? You talk about component stuff. You made a lot of tools that really help that. I would like to see that more often when you have teams that are going at a million miles trying to go from sprint to sprint.
Right. I think it's a great question. And I wish I had a better answer. It's sort of a project management philosophy, and, setting goals that are probably harder than we currently do, and giving ourselves time to hit them. You know, in the context of this fast-moving biotechnology, it's kind of hard to do that. So I think it's a very fair question, and I would hope someone else could answer that's on the call, because I don't have a good one.
Some of the, at least the stuff that you show for packages of managers would be very helpful just to get a beat on what everyone else is doing. But yeah, it's sometimes requires a very strong hand in order to make sure everybody's doing their homework.
Yep, we're learning about that. Late in life, I might add.
Thank you.
Yeah.
All right, other questions? I see that Nitesh made a shout out to the Bioconductor Slack channel, which I can only support. It is my source to go to with whatever technical questions. I typically have a very active channel and a lot of helpful people on there, which are always happy to discuss problems around R and Bioconductor. Any other questions for Vince?
I'll stop sharing. Maybe we can all see each other.
If not, I say thank you so much, Vince, for coming over and for a great talk, for taking the time. I know the new release is coming up, so I think it gets very busy these days. I conclude maybe with saying next week's seminar will be on spatial omics again, where we will have Artem Sokolov talking about the MC micro pipeline that is developed here at HMS. All right, then thank you very much, everyone, and maybe see you next week, and have a great rest of your day.
Thank you. Bye-bye.
Thank you.
Thank you.
Bye.